Hi, my name is Amanda, and today I'm going to be explaining excitation-contraction coupling. The first step takes place in the brain and the spinal cord. The brain and the spinal cord will send a motor neuron to go down into the skeletal muscle fibers to tell the body that there's going to be a muscle contraction. To start off, calcium ions will enter the axon terminal. Once the calcium ions enter the axon terminal, this will trigger the release of ACH. ACH is commonly known as acetylcholine. ACH will then bind with the nicotinic acetylcholine receptor. This change is picked up by the motor end plate, and this causes the ligand-gated ion channel to open and release sodium and potassium ions. This shift in the chemical balances causes a voltage change, and this is forming what is called an end plate potential, also known as an EEP. This change in the end plate potential creates what is called an action potential. The action potential will then travel down into the sarcolemma. Once it's in the sarcolemma, it travels down the T-tubule and then spreads itself into the sarcoplasmic reticulum. As we look closer, we see there are two different types of receptors. One receptor is called a dihydroiodine receptor, and that is found on the T-tubule. The second receptor is a radiodine receptor, and that is found in the sarcoplasmic reticulum. Once the action potential comes down the T-tubule and spreads into the sarcoplasmic reticulum, these two receptors are notified and release calcium. This calcium is released into the sarcoplasm. When breaking down muscle fibers more, we find there are two different types of filaments. The first type of filament is called a thick filament and is contained mostly of the protein called myosin. The second filament is a thin filament and it is called actin. On the myosin protein, we see there is a myosin head, an ADP molecule, and a phosphate molecule. On the actin protein, we see there is another chemical called tropomyosin and then another one called troponin. Both troponin and tropomyosin are very important in the interaction between actin and myosin. Since the sarcoplasmic reticulum released its calcium into the sarcoplasm, the calcium ions that were released will now attach to the troponin molecules. When this happens, the tropomyosin will move, and that reveals the active sites on the actin, so the myosin heads will be able to attach to the active sites. As we can now see, the myosin heads have attached to the active sites on the actin. We can also see that the myosin heads no longer contain that single molecule of phosphate. This is because when the myosin heads move to attach to the active sites on the actin, it uses the phosphate molecule to do this process. Once the myosin head is attached to the active sites on the actin, it forms what we call a cross bridge. Once the cross bridge is formed, the myosin heads will then demonstrate a power stroke. A power stroke is when the myosin heads will pull backwards because it will pull the actin protein over the myosin. We will later see that instead of the ADP that is attached to the myosin head, it will turn to ATP. When it turns to ATP, this will make the myosin heads release from the active sites on the actin. Since the ATP molecule attached itself to the myosin head, we can see now that the myosin heads have returned back to their original position. The ATP molecule is broken down back into the ADP molecule and the single phosphate molecule that we saw in the beginning. Altogether, this is what we call the contraction cycle. This cycle will continue as long as calcium is still present. Once our muscles are done contracting, the calcium will be transported back into the sarcoplasmic reticulum.